Okay, so good afternoon, all of you, and welcome to the next practical in TYBSc Botany. And the next practical is actually a problem which is based on 3-point cross or a cross between three genes. What is a 3-point cross? 3-point cross means there are three genes involved. There are three genes involved in the crossing over. Name of the scientist was T.H. Morgan. T.H. Morgan discovered that during the Lake Packetin or the zygotine substage of prophase 1 of meiosis 1, crossing over occurs between non-homologous chromosomes. One chromosome will cross over to other chromosome. Thus, there is exchange of genetic material. Because of crossing over, there are different shapes produced, which are called as chiasmata. Further, it was suggested that on the basis of crossing over, something which is called as chromosomal map, a chromosomal map was discovered. Now, what is a chromosomal map? A chromosomal map is a map that will give you the location of the genes, that will give you the location of the genes and distance between them. For example, if we study a map of Maharashtra, what we normally see, we see where Mumbai is located, and then we see where is Pune, where is Nasik, where is Nagpur. Then what we say, Pune is on the left hand side of Mumbai and Nagpur is on right hand side of Mumbai. That is called as location. So as we study location, even in chromosomal mapping, you can study that which gene is located where. This is called as location of the gene on a particular chromosome. For example, there are three genes A, B, C. We know that B is in the center, A is on the left hand side of B, and C is on the right hand side of B. But if there are genes, for example, WAH, three genes I'm giving you, WAH, then how can you find out the location? There is a method to find out the location of the genes. The method I'll be teaching you today and in the next practical, which I'll be taking on 3-point cross. So, what does a map normally gives? It gives you the location. And second thing, what is present in the map? Distance between the 2 points. For example, distance between Mumbai and Pune or distance between Pune and Nasik. Likewise, even on a particular chromosome, when the genes are present, you can find out distance between any two genes. For example, distance between A and B, or for example, distance between B and C, or if the genes are random, distance between gene W and gene A, or between any suitable genes. So when you find out distance between any two genes, it depends. It depends on the percentage of crossing over. What is crossing over? One chromosome will cross over with another chromosome. So the percentage of crossing over between any two genes is equal to the distance between any two genes. This is the formula. Distance between any two genes on a particular chromosome is equal to distance between them is equal to percentage of crossing over between any two genes. Now, when we say percentage of crossing over, it is of two types. It can be single crossover. Short form is SCO, or it can be double crossing over, DC and O. Next point to tell you, as you know that when we write distance between Mumbai and Pune, what we write? Mumbai and Pune are 167 kilometers apart. So there is a unit for writing the distance. In the same way, the unit for writing distance between any two genes or a chromosomal map or a genetic map, the unit is called as map unit. Unit between, I'm sorry, distance between any two genes is donated by the unit which is called as map unit, or it is also sometimes called as CM, that is centimorgan. Now, this is in honor of the scientist T.H. Morgan, who discovered chromosomal mapping. As I told you, in diploid organisms, the 3-point cross will involve 3 genes. If the problem is given on 3-point cross, you'll be asked 2 things. One, to find out location of the genes. How to find out location? You have to find out the percentage of single crossover plus double crossover upon total into 100. Second thing, you have to find out the location of the genes. So with this short introduction, now I will be dealing with a problem, a 3-point cross problem. I'll just show you the problem now. Yeah. Is the is it clear on your screen? Can you see the problem properly?
No,
Somebody
ma'am.
not able to see? Okay, just a minute. Now?
Yes, ma'am.
Yeah, okay, thank you. Should I enlarge it a little bit? I'll just try. This problem is already put in your Google Classroom. Today you'll be writing this problem in your journal, and we'll have one more revision in after 2 or 3 practicals. Yeah, I think it is very clear now. Everybody can see this? Yes?
Yes, ma'am.
Okay, thank you. So this is the first problem. I'll be taking 6 to 7 problems. First problem I'll explain here, and 6 to 7 problems I'll give for revision later on. So the problem is a 3-point cross problem. This is compulsory in your practical exam. Either you will get a problem or mutation, or you'll be given a problem and you have to construct a chromosomal map. So this is the problem given, and this entire thing is a problem. And what is asked? Construct a chromosomal map. So before going with the problem, let me tell you what is the chromosomal map. Yeah. The 3 genes mentioned in the problem are W, A, and H. And this, what has been constructed, this is called as chromosomal map. Now in this, we see that gene A is in the center. How to find that? I'm going to teach you right now. Further, distance between W and A, how much? Distance between A and H, how much? I've left it for you to do it in your journal later on. First, let me explain you. A problem is given in Drosophila, an individual with red eyes. Red eyes. Gene for red eyes is W. Hairless body. Gene is capital H. And anthocyanin pigment. Gene is capital A. So this is one individual, capital W, capital H, and capital A. This is crossed with another individual having white color of the eyes, gene small w, hairy body, gene small h, and anthocyanin less, gene small a. This individual is crossed with this individual. The results obtained are given below and construct a chromosomal map. Now, what are the results obtained? The offsprings which were produced showed all these combinations. Which were the combinations? Combination number 1, red, anthocyanin-less, and hairless. This combination was 349. 349 individuals were showing this combination. Similarly, all other combinations, white, anthocyanin, hairy, 12. Red, antholess, hairless, 14. So these were the actual individuals which were obtained. So the problem will be given to you in this manner. And what is asked? Construct a chromosomal map. Now here you have 3 genes, W, H, and A. They are dominant and one is recessive. First thing you have to find out which gene out of these 3, W आहे, H आहे, आणि A आहे. First solution will be to find out which of the 3 genes is in the center. Once you find out which gene is in the center, the second part of the solution will be to find out distance between them. Because unless and until you finalize the location of the gene, you cannot find out the distance between the successive genes. So problem will be given like this. Problem will be very much similar, only the figures, these figures, and the names of the genes will change. All the other things will remain same. How to solve it? See here. Solution. As I told you, there are two solutions, solution 1 and solution 2. In solution 1, you're going to find out which of the given genes is in the center. मध्ये कुठची जीन आहे त्याला म्हणतात finding out location of the gene. For finding out location of the gene, you have to arrange the data in the descending order. Arrange the data in the descending order so that you can find out which are the parental types, which are the single crossover types, and which are the double crossover types. So the above data which is given, I have arranged the data, only the data is arranged in the descending order. What is descending order? The most at the top and the least at the bottom. जे सर्वात जास्त आहेत ते वरती आहेत and that is reducing. Now here there is a slight modification. You can have 44 and 41. Only the small difference. This should be here and this should be here. Make that change. 44 should come first, which is followed by 41. Okay. Rest always okay. So arrange the given data in the descending order to find out parental type, SCO and DCO. Now, after the data is arranged in the descending order, the two types with maximum numbers, which are they here? Red, anthocyanine, hairless, 349. And the second highest is also 349, white, antholess, hairy. So number 1 and number 2 are parental types. They are parental types. Means the individual with red anthocyanin pigment hairless is crossed with white antholess hairy. The numbers which you get with maximum number in the data 349 and 349. This will be called as parental. After you arrange in the descending order, सर्वात जास्ती दोन जे असतील वरती पहिले number 1 and number 2, they will be called as parental types. Similarly, सर्वात कमी दोन जे असतील, the least ones, 14 and 12, these two, they are called as double crossover types. सर्वात जास्ती they are parental आणि सर्वात कमी जे आहेत they are double crossover types. Now what will remain? You will get 4 inside, 44, 41, 46, 45. 4 will remain in the center and they are single crossover types. They are single crossover types in region 1 and region 2. Region 1 means between gene 1 and 2 and region 2 means between gene 2 and 3. So after arranging the data, these two are parental, these two are single crossover, and 4 in between are double crossover types. We move on to the first solution, solution 1, to find out location of the genes. Now we don't know which gene is in the center. So we have to assume one after other. If your assumption comes correct in the first slot only, you need not do case number 2 and case number 3. There are 3 genes, so there can be 3 cases, either A in the center, H in the center, or W in the center. One by one we'll try. Case 1, assume that gene H is in the center. So how to write it? Capital W, capital H, H मध्ये, and A. Small w, small h, small a. Now, we have to show a double cross. How the double cross will go? Capital W, small h, capital A. I've written here. Capital W, small h, capital A. And second will be small w, capital H, and small a. So, which types you get? You get red, hairy, and antholess. And you get white, hairless, and antholess. Now, these two types, double crossover types. Compare this double crossover types with these double two. What is the combination here? Here the combination बघा काय आहे, रेड बरोबर अँथोलेस आणि हेअरलेस आहे डबल क्रॉसओव्हर मध्ये. इकडे आहे का रेड बरोबर हेअरलेस आहे? नाहीये. रेड बरोबर काय आहे? हेअरी. सिमिलर व्हाईट बरोबर कॉम्बिनेशन काय आहे बघा? व्हॉट इज द कॉम्बिनेशन? व्हाईट बरोबर अँथोसायनन आणि हेअरी. हिअर व्हॉट यू आर हॅव्हिंग? व्हाईट आणि हेअरलेस. सो what we have assumed is not correct. Why? The double crossover types here do not match with the double crossover types given in the data. Therefore, gene H is not in the center. Therefore, gene H not in the center. Case number 2. Assume that gene W is in the center. So how you will get? Capital A, capital W, capital H. Small a, small w, small h. Show the double crossover, capital A, small w, capital H, and small a, capital W, small h. Now see the combination बघा. Antho, white आणि hairless. White बरोबर antho आणि hairless आहे. बघा का? White बरोबर antho आहे. Hairless आहे का? नाहीये. सेकंड बघा रेडचं कॉम्बिनेशन बघा रेडचं. रेड बरोबर काय आहे? हेअरी आणि अँथोलेस. Compare that. Red borough hairy. Antholis is there, but hairy is not there. So once again, we can say that these double crossover types are not tallying with the double crossover types you get here. So what is the conclusion? Conclusion is that the DCO types do not tally with our data. Therefore, one line you write here, gene W is not in the center. You can write it here. What to write? Therefore, gene here, gene W, sorry, not in the center. Now, only one option is left out. And what is the option? बघा इकडे आपण बघितलं H सेंटर मध्ये नाहीये कारण टॅली होत नाहीये. W पण नाहीये. So what is the option left out? A. Assume that gene A is in the center. So what you will have now? You will have capital W, capital A, capital H, small w, small a, small h. And you show the double cross and you get the combination red, antholis, hairless. Let us check out red, antholis, hairless. If the combination is there in our descending order. Red, antholis, hairless. See, it is matching. And the second one will also match. इकडे काय second one? White, anthocyanine, hairy. Let us see in case number 3. Yes, see it is here. White, anthocyanine, hairy. So these types are matching with the types in our data. Why are they matching? They are matching because gene A, our assumption that gene A is in the center is correct. So whenever your assumption is correct, the double crossover types here will tally with your double crossover types given in the data. So what is the conclusion? The DCO types tally with our data. Therefore, gene A is in the center. Gene A is in the center. Now, supposing by default or by fault, if you have taken A only as case 1, जर तुम्ही case 1 घेतली असती A in the center and you get the correct data, you did not do case 2 and 3. तुम्हाला कळतं मी काय सांगते? जर तुम्ही पहिलंच assumption A घेतलं असतं, A in the center, तुमचं टॅली झालं असतं. In that case, do not do case 2 and 3. When you get it correctly, don't proceed further. If you don't get it correctly, you have to proceed further. So this is the first step. And what is the first step? To find out location of the genes. So what location we have got? We have got capital W, then we have small a, small h, small w, small a, small h. So this is the location of the genes. Half part of the portion is over problem. Now we move on. What is the next step you have to do? You have to find out distance between W and A and distance between A and H. Here and here. This will remain same. Distance between W and A or distance between A and H. This will do by help of the numbers which are provided here. Let us see how it goes. Yeah. Solution number 2. To find out distance between any two genes, the formula is single crossover plus double crossover types. I repeat, single crossover types plus double crossover types upon total into 100. This will give you the percentage of crossing over. This will give you the percentage of crossing over between two genes, and the percentage of crossing over between the two genes equals to the distance between the two genes. So you know that here now. First you will find this and then you will find this. This is called as region 1. This is called as region 1 and this is called as region 2. First we'll find distance here and then distance here. बघा पहिलं solution to A. Distance between W and A. For that, what you have to find out? You have to find out SCO between W and A. DCO you of course know. So how to find out SCO? See here. Cross is between W and A. This is single crossover. बघा इकडे single crossover between W and A. See how we have shown. Single crossover between W and A. After crossing over, what will happen? You will get the combination capital W, small a, small h. These are the genotypes. Capital W stands for red color, small a stands for anthocyanin-less, and H stands for hairy. And if you see this combination, from here you will get that this is 41 in number. This is 41 in number. Similar way, the second one, the second single crossover, small w stands for white, capital A stands for anthocyanin, and H stands for hairless. In the data, this combination is 45. Now, single crossover you can find between these two genes, and that is 41 plus 45. What about double crossover? Double crossover will be same for all the genes. And how much it is? I repeat, double crossover types will be same for all the three genes. Double crossover types are 14 and 12. They are 14 and 12. They will remain common for all the three genes. Now, formula, single crossover between W and A. How much you have? 41 plus 45 plus double crossover, 14 plus 12 upon 900. आता हे कुठून आलं 900? See here. सगळ्याची total करा. Total all this. Total will be 900. Total offsprings, if you calculate, add all this, number you get is 900. Already I've done for you. Total is 900 into 100. So calculate this and write in your journal. You'll get some answer. 112 upon 900 into 100. Whatever answer you get here, this is the distance between W and A. Supposing I get 9, I get the answer as 9. Okay, then the distance will be between W and A will be 9 MU. That is 9 map units. You have to put it here, and this is the result. This is the result. Is there anybody? Hello, hello, dear students. Hello, आशिष. हाँ,
Yes,
तुमच्याकडे
ma'am.
कॅल्क्युलेटर असेल तर जरा मला सांगा किती आहे. कॅल्क्युलेट करा. 112.
12.44.
How much? 12.
12.44.
Is it correct? I've written. बरोबर
Yes,
लिहिलंय?
ma'am.
See here. 12.44. What is the unit? Map unit. Fine. जसं आपण आता पहिल्या दोन जीन्सचं केलं, we have seen between W आणि A. तसंच आता आपल्याला करायचंय between A आणि H. मी इकडे क्रॉस दाखवला नाहीये. Between A and H. We have 44. हे जरा टोटल करून सांगा मला. आन्सर
12.88.
12 point how much? 88. Now 12.44. You have to write here in between. इकडे पण तेच आहे. This distance between W and A is 12.44 map units. And distance between A and H is 12.88 map units. You have to write it here between the arrows. So this is called as a chromosomal map. This is called as chromosomal map. Now in this, I have not shown the cross here. You have to write on your own. Here you have to show a cross between A and H. Cross इकडच्या ऐवजी कुठे दाखवायचं तुम्हाला? Between this and this. And then write the genotypes, write the phenotypes. You will get 44 plus 46. That thing I have not written here. Please do it in the journal. So this is the problem number 1, 3-point cross. Find out the chromosomal map.